Dear students, once again I'm meeting you to discuss more about the microbes in human welfare. And this lesson, we have the last part, that is what is bioremediation. Bioremediation. And before starting that one, the last class I mentioned about, that is the bacteria which are acting as biofertilizers. So we had talked about biofertilizers. I mentioned about one of the bacteria, Acetobacter. Now, the nitrogen fixing bacteria is Acetobacter and not Acetobacter. I mentioned about what is called Acetobacter. This is not Acetobacter, but it's Acetobacter. Acetobacter acid is responsible for the production of acetic acid. Now, the Acetobacter is a free-living bacterium responsible for fixing atmospheric nitrogen, and it is not Acetobacter. Make a correction. That's a made mistake. Now, let's go for bioremediation. So, what is bioremediation? Cleaning the environment with biological options. It's a simple term. Cleaning the environment by biological options. But as per the book, what is the definition? So, here, the use of naturally occurring microorganisms or genetically modified or engineered microorganisms which are used to degrade or to reduce the pollutants is called bioremediation. So, use of naturally occurring microbes or genetically engineered microbes to reduce or degrade pollutants is called what is known as bioremediation. Now, cleaning the environment by using what is called the microbes. Now, actually when you are comparing what is called as bioremediation with any other remediations, now it's more advantageous. It is more advantageous. In what way? Because the bioremediation is less expensive and more sustainable than any other remediations available. That's why it's being adapted nowadays to reduce the pollution, because it's less expensive and more sustainable, that is, than any other remediations available. Now, the bioremediation is grouped under two headings, or it is being grouped under two headings. One, in situ bioremediation. Another one, ex situ bioremediation. In situ means you know that one, even for in situ and ex situ conservation of animals. In situ means we have, for example, I am saying in situ conservation of animals. The animals are conserved as such in their original place. That's called in situ conservation. For example, Mudamalai sanctuary. And likewise, ex situ conservation. The animals are actually brought and then a natural adaptation is being provided for maintaining the animals. That is called ex situ conservation. They are removed from the forest and a medium is maintained, that is in the environment made by human beings where the animals are kept. That is called ex situ conservation. Likewise here, there are two types of bioremediation. Another one, just what is called in situ bioremediation. Another one, ex situ bioremediation. What is in situ? The treatment of contaminated water or soil in the site where they are present as such, and that is called in situ bioremediation. The another one, ex situ bioremediation. Here, the treatment of contaminated water or soil that is removed from the site and then treated. It is not treated as it is there itself. They are being removed from that area and they are being treated in some other place. That is called ex situ bioremediation. These are the two groups of what is called bioremediation. The next one, what is the role of microbes, or how are microbes acting, that is in degrading the pollutants. So the microbes are degrading the pollutants either by means of aerobic method or by means of anaerobic method. So we can just take first one, action of aerobic microbes. How far they are useful in degrading the pollutants. The next one we have to go for action of anaerobic microbes. Okay, let us divide that one into two. Action of aerobic microbes. Another one, action of anaerobic microbes. So let's proceed now. Now let's see what are the microorganisms involved in bioremediation. There are two groups of microbes. One, aerobic aerobic microbes, another one anaerobic microbes. Now let's see what is the role of aerobic microbes, how far they're acting in degrading the pollutants. So number one, action of aerobic microbes. These microbes can degrade the pollutants in the presence of oxygen. That's why they're called as actually aerobic microbes. And they degrade mainly what we have, the pesticides and then hydrocarbons. Their main action is on pesticides and hydrocarbons. In our syllabus given 3 examples. Okay, all the 3 are bacteria. So number 1, Pseudomonas putida. This bacterium is a genetically engineered bacterium. It's not existing naturally, it is being modified through genetic engineering. So, these are genetically engineered microorganisms, or simply we can say GEMs. Genetically engineered microorganisms, it's a bacterium. One Indian-born American, he settled in America. He just obtained what we have, the patent. Just we can say, that one, the license like that. Called by name Ananda Mohan Chakrabarti. So he is an Indian-born American. Obtained patent for this bacterial strain. He only can use this bacterium for just what is called the clearing the oil spills. Now it is a multiplasmid, just actually hydrocarbon degrading bacterium. A bacterium having a plasmid. The plasmid is genetically modified in such a way that it has a number of genes to degrade the oil, to degrade the hydrocarbons found in the oil spills. That's why it's called multiplasmid. Now I am drawing a simple diagram of the plasmid. Now it is having the genes like opt gene, TOL gene, XII gene, CAM. These are all actually the genes responsible for degrading what we have the hydrocarbons found in the oil. Now what are these actually genes normally acting? So they are acting on what is called octane, the opt gene for the degrading octane, toluene, just TOL gene, and then xylene, that is XOL gene, and camphor, that is CAM gene. That's why it's called as multiplasmid, having number of genes responsible for degrading the hydrocarbons found in the oil spills. So these are all the main hydrocarbons found in the oil, like octane, toluene, xylene, and camphor. So they are actually spreading what we have what is called the hay, the straw. How can they prepare this one? So a tank with water containing this bacterium is immersed with what we have, the straw. So that what is happening, the water seeps into the dry straw along with what is called this bacterium. Then the straw is being dried. Now the straw, the dried straw, contains this bacterium. Wherever we have oil spills, that is leakage from the ship while transport from one place to another in the sea, they are spreading the straw. The straw contains, that one, the hydrocarbon just digesting actually the genes. So they are capable of degrading the hydrocarbons because the oil seep into the straw where you have actually the degrading genes are present. They can degrade what we have, the octane, toluene, xylene, and camphor, the main hydrocarbons present in the oil spill. This is a mechanism. It's not necessary for it. So the meaning for multiplasmid, having different genes for degrading different types of hydrocarbons found in the oil spills. So it is a multiplasmid hydrocarbon degrading bacterium having a plasmid, that is multiplasmid, having many genes that can degrade and digest hydrocarbons in the oil spills. So during transport by the ship, there is a chance of leakage of the oil. The oil is normally just spilling on the surface of what is called the ocean water. They can be degraded with the help of what is called this genetically engineered bacterium that is prepared or made by an Indian-born American, not American, Indian-born American settled there. He only prepared. He only has a patent to use this bacterium. So that's about number 1, aerobic bacterium, just which cause the degradation of oil hydrocarbons. Now the second aerobic bacteria involved in the degradation of PET plastic. Now you know the PET bottles. The PET bottles are nothing but thermophilic polymer resin. Now on the surface of which we have certain bacteria, they are growing to get their carbon source. And by that way they are removing the pollutants. Now one such bacterium is Ideonella sakiensis, a kind of bacterium. So it's growing on the surface of the PET bottles. I mentioned just the PET bottle is a thermophilic polymer. Polyethylene thermophilic acid, a polyethylene thermophilate. Polyethylene thermophilate. It contains poly, just actually polyethylene, as well as you have also thermophilic acid, the combination. That's why it's called as PET. Now, this Ideonella is currently tried for recycling of PET plastics. This bacterium produces two enzymes. One, PETASE, that is nothing but what we have polyethylene terephthalate hydrolase. Another enzyme, mono 2-hydroxy ethyl terephthalate hydrolase, that is called MHETAS. These two are responsible for breaking down what is called the PET plastic. Now in the diagram what is given, a small piece of what is called the bottle is exposed. That is what is called the PET film. This is the surface of the bottle. One small portion is exposed in this. Actually this is a piece of what is called the PET bottle. Now Ideonella sakiensis normally adhere to the PET film. That is the surface of what is called the bottle. What it normally does, it breaks down PET into two components. One is terephthalic acid, another one polyethylene glycol. While doing so, they are normally getting the carbon source for their what is called actually the fixation. So once they are being broken, now these bacteria, Ideonella sakiensis, found on the surface of the bottle, that is eat terephthalic acid and polyethylene glycol. In that way, they remove what is called the plastic, the PET plastic. So the recycling of PET plastic depends on mainly what we call this Ideonella sakiensis. This is a currently just actually tried method. This is because of the possession of two enzymes by the bacterium, that is namely Ideonella sakiensis. I mentioned these are all the bacteria. They are found on the surface of what we call the PET bottle. I am exposing only a portion of the PET bottle, that is called as a PET film. That's nothing but the surface of what we have, the PET bottle. First the Ideonella sakiensis adhere to the PET film, then it causes the breakdown of PET into terephthalic acid as well as what is called polyethylene glycol, and then they eat terephthalic acid and polyethylene glycol for their carbon source. This is what is happening. Now what is actually happening? Now I mentioned PET referring to polyethylene terephthalic acid. Now PETASE, polyethylene terephthalate hydrolase enzyme. Then MHETASE, mono 2-hydroxy ethyl terephthalic or terephthalate hydrolase. These are the two enzymes. Now what is happening? What actually first? Now this PET is being converted into mono 2-hydroxy ethyl terephthalic acid with the help of what is called PETASE enzyme, that is nothing but the hydrolase. Once MHET is formed, it is being hydrolyzed by another enzyme, that is mono 2-hydroxy ethyl terephthalate hydrolase. I will show the reaction now. Now the PET plastic is nothing but what is called a thermophilic plastic resin, that is highly resistant to heat. That's why it's being used also in ovens. Now the PET plastic, just normally what is called polyethylene terephthalate, that's the name of the plastic PET expansion. With the help of enzyme, the hydrolase enzyme for the breakdown of PET to form MHET. That is nothing but mono 2-hydroxy ethyl terephthalate. That is a second product. This is being broken further with the help of MHETASE, the hydrolase enzyme, into terephthalic acid and polyethylene glycol. They are being consumed by what we have actually the bacterium Ideonella for their carbon source. Now in the case of Pseudomonas foetida, what I discussed, the genetically engineered microorganism, how this it degraded. That is normally there is oils, that is from the oil spills, the hydrocarbons. Now there is a process of bioremediation. Now the microorganisms eat oil and other organic contaminants. So this is oil droplet, it's given in the form of bread. So these two diagrams represent the eating process. Then what is happening? The microbes digest and break down that oil into carbon dioxide and water. And finally the carbon dioxide and water being actually given off by the microorganism. This is a mechanism, simple mechanism. Consumption of oil, breakdown of oil into carbon dioxide and water, then giving of carbon dioxide and water from the microorganism. This is what is called actually the process of bioremediation that is happening by the activity of Pseudomonas foetida, the gem. Now the third example, actually aerobic bacteria involved in the biodegradation process, that is Nitrosomonas europaea. Now it is also capable of degrading something. What are the materials which are being degraded by this microbe? That is Nitrosomonas europaea. So it degrades benzene anaerobically and also oxidizes what we have normally the hydrocarbons like that is sorry, I made a mistake here. So these bacteria are capable of degrading benzene and a variety of halogenated, just like you know that one halogen compounds, bromine, ethylene, etc. So, and a variety of halogenated organic compounds, mainly trichloroethylene. So it is an aerobic what is called bacterium which degrades benzene and a number of halogenated organic compounds, mainly trichloroethylene. Okay, then what is the role played by actually the anaerobic microbes in the just degradation or bioremediation of the various materials? So that's why I'm writing here action of anaerobic microbes. So these microbes normally degrade the pollutants in the absence of oxygen, and that one just in the presence of oxygen they degrade. And here in the absence of oxygen anaerobically, these microorganisms degrade the pollutants. The number one, the bacterium, Dichloromonas aromatica. It functions dually. It oxidizes certain hydrocarbons. At the same time, just anaerobically, it breaks down or digests what is called benzene. So these are capable of degrading mainly benzene anaerobically. And also by oxidation process, they remove xylene and toluene, the hydrocarbons. So xylene and toluene are hydrocarbons, and we have the benzene, the aromatic, what is called the organic compound. That's why the name is given as aromatica. They are concerned with the removal of benzene. Now the second example, and this one anaerobic actually microbe which degrades pollutants, Phanerochaete chrysosporium. It's not a bacterium, it's a fungus. So it is an anaerobic fungus which has the potentiality of biodegradation of a variety of compounds. For example, you see that one, pesticides, polyaromatic hydrocarbons, then also trinitrotoluene, then cyanides, then carbon tetrachloride and dyes. So it is the ability of actually degrading a variety of what is called the compounds, both the pesticides as well as the hydrocarbons, etc. So that's about what we have the fungus. The first one is a bacterium, the second one is a fungus. We have also another bacterium, another fungus, two more examples to complete what we have the action of anaerobic microbes. Let's see now. The third example for anaerobic breakdown of what we have any pollutant, there is nothing but what we have in the form of bacteria, just normally or fungus. coming back to this one. So Dihalococcioides, a kind of bacteria. Now these bacteria degrade toxic, what's called trichloroethane, not ethylene, ethane into non-toxic ethane. Now the last one, Pestalotiopsis microspora. Pestalotiopsis microspora, it is a fungus. It is living within the plant body, hence called endophytic fungus. The meaning for that one, living within the plant body, endophytic fungus. It is capable of degrading and digesting another organic compound, what's called polyurethane. Polyurethane also being used in the preparation of just some plastics. And because of its activity, it is being considered as the most actively potential candidate for bioremediation or bioremediation project work with large quantities of plastics. So this is the most suitable candidate or a potential candidate for bioremediation project where you have large number of plastics are available, large quantities of plastics are available. There is the importance of what is called Pestalotiopsis microspora, a fungus. So in the anaerobic breakdown, you see that one, we have what is called, that is Dichloromonas aromatica, that is a bacteria. Then the second one, what we have, just we see that the panarochete, Cryosporium or Chrysosporium, panarochete, Chrysosporium, there is a fungus. Now this one, Dihalococcioides, a kind of bacteria. The last one we have just actually a fungus, Pestalotiopsis microspora. So two bacteria, two fungi forms are given in the book for the action of anaerobic microbes. They are working in the absence of oxygen. So with that I concluded the lesson, microbes in human welfare. We have to go for the next lesson, that is applications of biotechnology. You have already studied biotechnology in botany, the basic principles and processes. Now here we have to go for the applications. The production of different kinds of medicines or in the production of what is called cloned animals, like that. We'll see that one in that lesson. Okay, till then, goodbye. Thank you.